Tyrosine kinase receptors are important proteins involved in many cell signaling pathways. They play an important role in the regulation of cell growth and proliferation. Tyrosine kinase receptors are transmembrane receptors, with an extracellular portion containing the ligand binding site and an intracellular portion that contains the tyrosine kinase domain. When inactive, the pair of receptors are apart from each other within the membrane. The activation of tyrosine kinase receptors begins with an extracellular ligand binding to each of the monomers. The two monomers then come together to form a dimer in a process known as dimerization. Tyrosine kinase residues on the intracellular portion of the receptor hydrolyze ATP in the cell into ADP plus a phosphate group. Tyrosine kinases phosphorylate each other in a process known as autophosphorylation until all the tyrosine kinases have become phosphorylated. The phosphorylated tyrosine kinases can then recruit and activate relay proteins which activate proteins involved in signaling pathways. An example of this is when the VEGF ligand binds to a tyrosine kinase receptor, the downstream effects of which cause new blood vessel growth. This is just one of the many examples that use receptor tyrosine kinases. It is important not to confuse receptor tyrosine kinases for non-receptor tyrosine kinases. Non-receptor tyrosine kinases do not have a transmembrane domain. They are cytoplasmic tyrosine kinases that associate with a receptor that does not have kinase activity. For example, in the JAK-STAT signaling pathway, JAK has kinase activity, but the receptor it associates with does not.